I want to thank our audience really for joining us this evening, and also the team at Blood Cancer United, which has been a great partner. You'll hear more about them a little bit later from Nicole Fortoso, but we have had a long relationship with Blood Cancer United, formerly Leukemia Lymphoma Society. And it's great to partner on this program. And I know a number of the people in the audience also have those connections. I'm also really happy to be here tonight with my colleagues from the Perlmutter Cancer Center and NYU Langone Health. I joined NYU about 6 months ago. Dr. Diefenbach, who you'll hear from in a minute, was already here and really someone who I've been very excited to collaborate with, and you're also going to hear from a number of other recent arrivals. We have a lot going on in blood cancers at the Perlmutter Cancer Center and NYU Langone Health, and it's really exciting to be here with you tonight. And all the great things that are happening here with my colleagues and many others here at the center, some of whom you'll hear from in the other sessions around blood cancers. Very briefly, the American Society of Hematology is really one of the big national meetings every year where new data are presented. And so we are going to give you, this is where all the hematologists in the country and throughout the world come together to hear what's new. And we're going to have a summary for you of that tonight. And you can see our lineup. I'll introduce the speakers as we go. It's really great to have my colleague, Dr. Catherine Diefenbach, who is the director of the clinical lymphoma program here and associate professor at the Perlmutter Cancer Center, to give us some updates on aggressive lymphoma. She's someone who's been at the forefront of new therapies for various types of lymphoma, and is really a superb colleague and expert in many of the agents you'll hear about tonight. So we'll start with Dr. Diefenbach.
I want to thank you for that kind introduction. I want to thank you, Katie Grace, and also Blood Cancers United for setting up this amazing program. And most of all, everyone who is here on this webinar. I would be absolutely happy to talk to you guys afterward about any question you have in lymphomas. And if I don't answer you in the Q&A, my email is on this, and feel free to email me. So what I'm going to talk tonight about is updates from ASH 2025 in aggressive lymphoma. And let me just start by saying there has been an explosion of research in aggressive lymphoma, and I got 8 minutes. So I have to choose one thing to talk about you in aggressive lymphoma, because if I was going to tell you all the insights in aggressive lymphoma, it would take this whole webinar. So what I think is most important is the integration of bispecific antibodies into standard chemotherapy regimens. So a bispecific antibody is an antibody with 2 arms, or in the case of glofitamab, really 2.5 arms. And one arm grabs a tumor cell by a protein that sticks on the tumor cell. So that's the anti-CD20 arm. And CD20 is a protein that is very common on the surface of a tumor cell. The other arm grabs CD3, which is a protein that's on a T cell. And what it does, it takes the tumor cell and the T cell and it brings them next to each other. And it says, tumor cell, eat the T cell. T cell, eat the tumor cell. I'm very sorry. Tells the T cell, the cell of your immune system, to eat the tumor cell. By doing that, your immune system becomes activated and many more T cells then eat tumor cells. And so these are the 3 antibodies that have been explored in DLBCL and in low-grade lymphomas. They're going to come back in, I think, Dr. Martin's talk and Dr. Yamshan's talk in other contexts. But the 2 that are FDA approved in third-line DLBCL are epcoritamab and glofitamab. And then mosunetuzumab is approved in low-grade lymphoma, but was looked at and is under investigation also in aggressive lymphoma. So those are the 3 that we're going to talk about tonight, and they really make a tremendous advance in integrating immunotherapy into the treatment of DLBCL, which had previous to this primarily been treated with chemotherapy. So DLBCL, or diffuse large B-cell lymphoma, is the most common aggressive lymphoma. Cure rates, although we would like to cure everybody, we're particularly not good at curing elderly patients. And this management of the frontline treatment of DLBCL in the frail and elderly remains a strong unmet medical need, because we need to balance curative intent chemotherapy with mitigating toxicity so that we can maximize cure without giving a regimen that's too toxic to the patients. So there were 3 studies that were presented at ASH that integrated bispecific antibodies into frontline DLBCL for elderly patients. Two went into chemotherapy platforms, and one was a standalone for very frail patients. And that's what I'm going to talk to you guys about tonight. So the hypothesis is that highly biological active anti-lymphoma agents can overcome the fact that the unmet need of not being able to actively treat chemotherapy. So this was a regimen of polatuzumab, which is a chemo on a stick, uses CD79B to put chemo into the cell. Glofitamab, which I talked to, is the bispecific antibody, and then Rituxan, which is standard in all frontline chemotherapy regimens, is an antibody to CD20 also. So this was a regimen of glofitamab, polatuzumab, and Rituxan. And the study design was that basically patients with newly diagnosed lymphoma got basically polatuzumab and obinutuzumab, and they slowly went up on the bispecific to mitigate toxicity. They then got 6 cycles of everything together, and then they got 6 more cycles of the bispecific by itself. About 80 patients were treated. It was a one-arm phase 2, and it was 30 centers in Europe. These were all untreated patients who were greater than 60, and they could not be eligible for full-dose chemotherapy. But what's interesting about this is there was no chemotherapy in this. So the big worry with bispecific is this thing called ICANS or CRS. This is neurotoxin blood pressure toxicity with bispecific antibody therapy. And you can see that it was very low, so that there was almost no patients who had high-grade CRS, and it all really occurred in cycle 1. And same with the ICANS. Almost all patients had only cycle 1 and only grade 1 to 2. Infections were also quite low and very standard, similar to how you would be getting infections on chemotherapy. So there were no new safety signals. Next slide, please. What's really exciting is the response rate. So by the time patients had finished cycle 12, 81% of patients were in a CR. So an overall response rate of 90%. And 81% of patients were in a CR. And the patients who are in a CR have the best chance of being cured of their lymphoma. So that means basically almost all the patients who were treated with no chemotherapy had an excellent response. Also, what was really interesting is that not everyone in cycle 6 had all their cancer go away, but by more bispecific antibody, some patients converted from having a partial response to a complete response. And we can also see, and we'll just skip over this, that most patients who had a complete response remained in their complete response, and that patients at 1 year mostly did not relapse. So this is a huge advance because there has been really no regimen of non-chemotherapy that has been able to control disease in patients who were not eligible for chemotherapy. This study was slightly different. These were slightly fitter elderly patients who got something called mini CHOP, because CHOP chemotherapy can be given to older patients who are fit in a mini version where they get 50% dose reductions in cyclophosphamide, vincristine, and doxorubicin. And they had a bispecific antibody called epcoritamab added. Again, the complete response rates were extremely high, very similar to what was seen in the prior studies, or slightly higher, and patients responded very fast. These patients only got 6 cycles of therapy. But they did get more chemotherapy up front. Next slide, please. And again, these patients, this is 2 years of follow-up now. Almost all the patients who had a CR, keep going. Almost all the patients who had a CR were in remission at 2 years. Next slide. And if you're in a remission at 2 years with large cell lymphoma, you're most likely cured. So this was a huge advance for the treatment of patients with elderly lymphoma who are fit for chemotherapy. Again, this was quite safe, with most of the safety signals no difference than what you would see on standard chemotherapy. And the CRS and ICANS, again, was very low, much lower than what you see with CAR T cells, and really only seen in the first full dose and not an issue subsequently. So finally, and I'll go very fast through this, for patients who are super frail, this was a study of just bispecific, no chemo, no polatuzumab, no Rituxan. So patients only got a bispecific, and this bispecific is not one that's approved in large cell lymphoma, but it said for patients who are really, really frail and can't get anything, how do they do if we just give them a bispecific? Next slide. And you can see that the response rates were actually, although not equal to the other studies, still very good for thinking you're only getting a single agent of one drug with aggressive lymphoma. So more than half of patients responded. And these were the most frail patients with the most aggressive lymphomas. And patients who had a complete response, again, next slide, did very, very well. So you can see that even for elderly, frail, unfit patients, many of them are getting disease control just with a bispecific antibody, and the toxicity is very low. So in conclusion, all three studies demonstrated impressive disease control and manageable safety with integration of bispecific antibodies into the frontline therapy for the frail elderly. EPCO-R-mini-CHOP is a full chemotherapy regimen, just mini, including anthracycline at lower doses. R-Pola-Glow has no cytotoxic chemotherapy. You can't do cross-trial comparison, but these both look highly effective. The Morning Sun is bispecific alone. It doesn't look as good as the other two, but it's much less toxic, and it was given to the frailest most elderly patients. How to make sense of this data moving forward? None of these regimens are FDA approved in the frontline. They all need to be validated in larger studies. For fit elderly patients, both frontline studies have similar data. Is chemotherapy necessary with bispecific or Rituxan? For unfit elderly patients, R-Pola-Glow is promising. For fit elderly patients, our mini CHP with EPCO would probably be, if approved, the way to go. And for the frailest patients, you can follow Morning Sun. I just want to highlight that we have actually participated in many of these trials, and that there are many exciting developments in DLBCL here. We have participated in a major way in frontline DLB bispecific trials, in trials for relapsed B-cell lymphoma. Right now, we have very exciting new bispecific antibodies targeting things like CD22, CD19, and soon BAFF-R, as well as B-cell degraders. And if you want to learn more about lymphoma research and clinical trials at NYU, my email is on the bottom of the screen, or you can reach out to me in the chat, and I'd be happy to talk to you and take your questions later. Thank you.
Thank you so much, Dr. Diefenbach. And I'll just mention if people have questions, feel free to put them in the Q&A. Some of them we'll answer through the chat function, some of which we'll come back to at the end for the group, depending on how generalizable they are, etc. And now we'll move to Dr. Sam Yamshan, who is the medical director of the Cellular Therapy Service. He's been doing a lot of great work around CAR T cells and cell therapy, as well as lymphoma in general. And it's great to have him give an update in this area. Thank you.
Thanks so much, Dr. Leonard. And again, thank you all for being here. And we're excited to share some of this data with you. Like Dr. Diefenbach, I felt overwhelmed by the task of having to cram all of the very exciting updates into 8 minutes, but we will do our best. I'm going to speak a little bit about some of the exciting innovations in CAR T cell therapy for lymphoma for ASH. And next slide, please. And these are my disclosures. But first, I think it's important to understand what exactly CAR T cells are. And CAR T cells, when I explain it to people, I think many people's first reaction is that it sounds like something out of science fiction. And in many ways it is. And essentially what we're doing with CAR T cells is actually training the normal immune system to become the special forces that fight lymphoma. And the way that works is that under normal circumstances, our immune system actually tries to fight lymphoma, but lymphoma cells are very smart and they find ways to hide from the immune system. And so what we're now able to do is we're able to take patients' own T cells out of their body, and then we send them off to a company where they're trained to be able to recognize and kill lymphoma. I call this step boot camp. And they become the special forces that are able to recognize the lymphoma even when it's trying to hide. And then we put the cells back. And because they're part of patient's own immune system, they're able to expand rapidly and to kill lymphoma cells. And they are really remarkably effective at doing so, and have dramatically changed the landscape for the treatment of lymphoma in the last few years. And so just kind of understanding how that works, what we do in order to make the CAR T cells work is we put this new protein into the surface of the T cells. And the way I kind of think about it is that protein has two parts to it. There's the lymphoma detector, or the chimeric antigen receptor that actually allows the T cells to recognize a designated spot on the lymphoma cells. And it's important that that spot is on lymphoma and not on other things. And then there's the costimulatory domain, which I like to refer to as the gas pedal that allows those cells to ramp up. So now with CAR T at ASH, I would put some of the innovations into three distinct categories. And I would say that we now have more access, new tools, and new combos. So CAR T moving into new diseases, enhancing existing CAR T cells, and new combinations with CAR T. So let's start by talking about new diseases. So I think one of the things that was most exciting coming out of ASH, and actually we got news from the FDA literally the day before our conference this year, of a new approval. So Lysocell, or Breyanzi is the trade name, is a CAR T cell therapy that's been approved in actually four other subtypes of lymphoma for the last few years, but is actually now the first CAR T cell that's approved in marginal zone lymphoma, which is an indolent or slow growing subtype of lymphoma. And this was based on data from the Transcend study, which we actually had access to before ASH, but got some updates. And just to quickly run through this data, I think what was exciting was that in patients who had seen many previous lines of therapy, there was a response rate of well over 90%, 95%, with about two-thirds of those patients having a complete remission on their scans. But most importantly, which is especially important in slow-growing lymphomas, many of these responses were quite durable, with a 2-year median duration of response, meaning how long the lymphoma stays away, of almost 90%. So both effective at shrinking things and keeping it away. Another CAR T moving into new spaces was for CAR T for treatment in Burkitt lymphoma. And Burkitt lymphoma is a relatively rare subtype of aggressive lymphoma, but its hallmark is its aggression. And unfortunately, up until recently, many of the treatments that we have for patients who relapse after chemotherapy have proven somewhat ineffective. And so this was a very small study, but one that was exciting for a lot of folks. That 10 patients received Brexucell CAR T, trade name Tecartus, which is approved currently in mantle cell lymphoma, but had a 100% response rate, which in a disease as aggressive as Burkitt lymphoma is very exciting. But also, again, thinking about that durability, 4 out of 5 of those patients who had a complete remission remained in complete remission at the time of study cutoff, which was very, very exciting. Again, very early days, but another example of CAR T moving into a new space. And, as Dr. Diefenbach mentioned, for patients with diffuse large B-cell lymphoma, which is the most common lymphoma, the current treatment paradigm is chemoimmunotherapy. But there are now a couple of studies, including one that was presented initially at ASH, of actually incorporating CAR T as part of the frontline treatment, trying to reduce the amount of chemotherapy given, particularly for folks where some of the responses to chemotherapy were suboptimal. So using a CAR T called YTB, which is a next generation from CAR T from Novartis, for patients with newly diagnosed diffuse large B-cell lymphoma, they saw both high response rates with an overall response rate of 89% and complete remission rates of 75%. So and some of these did appear durable. Again, it's relatively early days, and in a disease like diffuse large B-cell lymphoma, that durability really is critical. But it's exciting to see CAR T moving into new areas. So also important is, CAR T is very effective, but many patients do still relapse following CAR T. And so we are working to improve the CAR Ts that in diseases that we already have CAR T cells for. So I think very important is understanding how lymphoma gets away from CAR T. And I would kind of put them into 2 broad categories. One is that the CAR T cells get tired out over time, and the lymphoma is kind of able to wear them down, or that lymphoma is able to hide. And so, that CAR T, that lymphoma detector protein needs to be able to recognize a specific protein on the surface. And so sometimes lymphoma cells just stop expressing that protein. And so the lymphoma is then able to hide. And so a lot of our strategies are designed around mitigating both of these methods or of escape. So one exciting trial was the Atalanta-1 study, which was for mantle cell lymphoma, which was a study that selected for memory T cells. And those are T cells that are less prone to exhaustion. And so when they took out the T cells from people's body, they actually specifically selected memory T cells and only made CAR Ts out of an enriched memory T cell product. And what was exciting in mantle cell lymphoma was that, again, a CAR T where traditionally disease outcomes in later lines of therapy have been tougher, we saw really, really impressive overall response rates of 96% and complete remission rates over 90%, 92%. So, and of the patients who actually were infused with the CAR T, every single patient responded. And so this was very, very exciting. And importantly, looking at some of the markers of durability, again, early days, the patients did remain in remission. And this slide here from that study is just highlighting over here on the left many high risk disease features. So these weren't just they weren't just selecting for the easiest lymphomas to treat. Many of these mantle cell lymphomas had particularly high risk disease features and still had very impressive long-term remissions. Another strategy is to choose a new target. So currently all FDA-approved CAR T cells target a protein on the surface called CD19. But many lymphoma cells also express a protein called the BAFF receptor. And importantly, that BAFF receptor is not present on other normal cells, so that the CAR T is only attack the lymphoma, but not some of the other normal cells. So there was a small phase 1 CAR T study that was presented at ASH of the BAFF-R targeted CAR T cells, again, that showed a very, very high response rate, and that many of these patients, even a couple of years out, remained in remission with no patients relapsing. So very, very exciting results. Again, small study, early days, but we're certainly excited about this. So this just kind of summarizes, there's way more data than we could have possibly shown, but there are many, many different strategies to improve both CAR T cell fitness as well as attack different spots on those CAR T cells to prevent them from escaping the immune system. And so many, many, many different strategies than the ones I've shown being investigated right now. So just wanted to also highlight one study from our group. So we work very closely with laboratory collaborators, and one of the scientists that actually recently just joined us together from our old institution, Wendy Begelin, had shown that there is a protein called EZH2, an enzyme called EZH2, that if you inhibit it, kind of attacks the lymphoma at all these different angles. It both increases the fitness of those T cells and prevents them from being exhausted, but also kind of forces the lymphoma cells to express some of those proteins that allows the T cells to see them. And so we hypothesized that giving this drug, both before we collect the CAR T cells and after they're treated, would enhance CAR T cells kind of in both directions. And so again, we gave the treatment both before and after, and then continued it to try to kind of turn those T cells into the Energizer bunny that just keep on going. So what we found with this study was that we had a 100% overall response rate across 3 different lymphoma subtypes: diffuse large B-cell lymphoma, mantle cell lymphoma, and follicular lymphoma, with a 77% complete response rate. And many of those patients, in fact, the majority, still remain in remission. So, we also did many scientific analyses on the blood from patients and on the actual CAR T cell product to help understand why we were seeing such high response rates. And again, kind of based on our hypothesis, we found that it did kind of turn those T cells from those exhausted versions into the more pepped up, excited versions that we know are so important for long-term efficacy. And so based on this, we actually are also looking at this combination in combined with bispecific antibody therapy in follicular lymphoma, using mosunetuzumab, that one of those drugs that Dr. Diefenbach just mentioned. And so this will be open at NYU soon, and we are very excited to have this here based on the science from our scientists. So again, to emphasize, there is a lot happening in CAR T research in many different areas. And so NYU is leading the way in all these areas. We have trials upcoming in new diseases, primary CNS lymphoma and Burkitt lymphoma. We have several of these innovative products open already and more to come. Current trials with bispecific CAR Ts, allogeneic CAR Ts, and memory phenotype CAR Ts. And then again, those rational combinations based on science that was developed with Dr. Begelin, who's part of our group here at NYU. So, um, all in all, I think a very exciting time in the CAR T land and in lymphoma research in general. A lot of very exciting treatments coming forward. Like Dr. Diefenbach mentioned, I'm also available by email. So please feel free to reach out by email, or if for whatever reason Twitter is your thing, I'm also there. So looking forward to connecting, and thanks for having me.
Thanks very much, Dr. Yamshan. Now it's great to have the topic of indolent lymphomas, another major category of lymphomas. I'm sure many people are particularly interested in this subtype. A colleague, Dr. Peter Martin, who's the co-director of the hematology program here at the Perlmutter Cancer Center in NYU Langone, is going to give us an overview of new developments there. Again, just to remind people, the chat or the Q&A, not the chat, but the Q&A is open, and feel free to put questions there, some of which we'll respond to by text, and others we'll talk to in the open session a little bit later. Thank you. Dr. Martin.
Thanks, John. So I will touch on these topics today, including bispecific antibodies that you've already heard quite a bit about. So a little bit about mantle cell lymphoma, a little bit about follicular lymphoma, and an exciting new type of drug that we're seeing move forward in lymphomas. So I think both Catherine and Sam alluded to the fact that lymphomas are complicated. The tumor is not just composed of tumor cells, but there are a whole bunch of different cells in there. And we're fortunate now, as Sam mentioned, to have several labs that are specifically looking at the interactions between these tumor cells and their microenvironment, including the Wendy Begelin's new lab, and another collaborator is coming. So we have an increasing understanding of the role of the immune system and the way it interacts with the follicular lymphoma cells, and why that's so important in the management of lymphoma in general, but especially in follicular lymphoma. Based on that interaction, we've known now for a while that there's a role for these bispecific antibodies. I won't dwell on the mechanism of action. I think Catherine already discussed that, but I'll just mention that we have two bispecific antibodies currently approved for follicular lymphoma, mosunetuzumab and epcoritamab. A third antibody on the far right-hand, glofitamab, is approved for large cell lymphoma, not follicular lymphoma. And on the left side, odronextamab is under review and will likely be approved for follicular lymphoma at some point in the future. Then there are literally dozens of other antibodies that are in development. It's hard to keep up with this field. So this looks at some of the side effects of bispecific antibodies. Catherine already mentioned this to some degree. And I put this up because it just sort of shows a little bit about how they're used. First of all, you can see that in every case, regardless of which one we use, we start at a low dose and we slowly escalate it over time. In general, as we get to the higher doses, there's a little bit more of a side effect. And that side effect is called cytokine release syndrome, which is basically like the feeling that you get when you have an infection, except in this case, the infection is the lymphoma and it's your immune system fighting against the lymphoma. But in all of these cases, as we escalate it slowly, we can mitigate the grade or severity of the cytokine release syndrome and keep it at a fairly low rate, meaning it's fairly tolerable. And in all of these cases, these medications could be administered entirely as an outpatient for most people. This is just an example of the efficacy of these drugs in people with very heavily pretreated follicular lymphoma. There are multiple other studies that are similar to this, but you can see that very high response rates in people who had not responded very well to prior therapies. The first time to response is typically within the first 1 to 2 months, so it's very clear that they're working quite quickly. And when they're working, they're often working for 3 years or longer to keep the lymphoma from progressing, which is quite a significant period of time given the relative tolerability of these drugs and how the patients who participated in these trials may not have responded well to their prior therapy. When we administer bispecific antibodies, despite the fact that they're effective and well tolerated, I think this is one of the key areas that we need to consider. We don't just administer them like we're giving IV saline or salt water. We do have to prepare for them, and so every facility where these are prepared goes through an extensive education process to ensure that everybody is prepared. Our pharmacies are prepared, our nurses are prepared, everybody who's going to answer the phone at 2 o'clock in the morning if somebody has a side effect is prepared. And this preparation is ultimately what makes these drugs as safe as they are. And we're fortunate to have several locations at NYU where this preparation has already taken place. And so we're able to give these drugs safely throughout the New York area. So once you have a drug that you know is effective and safe and is already approved, very often the next question is, well, how can we make this drug work even better? And very often that involves combining a drug with other drugs. Sam mentioned one with mosunetuzumab combined with tazemetostat. There are numerous others that were presented at ASH. I just highlighted a few that were presented, including with zanubrutinib, golcadomide, or rituximab. And in some cases, these were in people with previously treated follicular lymphoma, and some they were in untreated follicular lymphoma. And you can see that they still appear to be quite effective with high complete response rates and fairly low toxicity. But these are all small studies, and so these data need to be replicated in larger studies in the future. This was perhaps the more noteworthy of the studies that was presented at ASH, and this was in people with previously treated follicular lymphoma. R squared, which is lenalidomide plus rituximab, is a very common regimen in that population. And the experimental arm in this trial was lenalidomide plus rituximab plus epcoritamab. And you can see on the right side of the slide that the addition of epcoritamab significantly improved the overall and complete response rates, that's ORR and CRR, significantly, as well as significantly prolonging progression-free survival. So that as a result of this trial, the combination of epcoritamab and rituximab and lenalidomide were approved in people with follicular lymphoma having received only one prior line of therapy. So this is the first time that we're seeing a bispecific antibody in people with only one prior line of therapy. Not surprisingly, when we add multiple drugs together, we do get a small increase in side effects. And so this basically involves a discussion. Anytime we're talking to somebody with follicular lymphoma, we, as a team, talk about whether the side effect profile is appropriate and what we're likely to get on the other side of it. Survatomig is a new bispecific antibody that targets not CD20 but CD19. And at NYU, we've had trials with this drug. The interesting thing about this one, not only is the interesting efficacy with you can see here the very high overall and complete response rates, as well as good durability of these responses at 12 months, but also the side effect profile appears to be safer than the antibodies that target CD20 with almost exclusively only low severity types of cytokine release syndrome. And so we have more trials with this agent moving forward in the near future. I'll pivot quickly to mantle cell lymphoma and basically just say that, I've had this as a personal interest for over a decade, along with Dr. Leonard and I. We've been working to move new concepts forward, including getting rid of chemotherapy. And it's become very clear from this last ASH meeting that this is now universally true. The whole world is on board with the concept of moving away from chemotherapy. This was one regimen that was presented, and I'll show it here. It's zanubrutinib obinutuzumab and venetoclax, basically administered continuously for 24 cycles. And you can see basically this is what we call a swimmer's plot. You have to imagine each one of these lanes is one person, and you can see almost without exception that everybody continues to be on treatment until they reach the 24-month period. And then in this trial, they were able to stop treatment. And of those who stopped, many of them continue to do well. Although it's important to note that there's limited follow-up when this trial only 6 months after they stopped, so not a lot of time. I'll pivot now to chronic lymphocytic leukemia, which is an indolent non-Hodgkin lymphoma. And for the longest time now, for the past decade, we've had 3 different drugs approved: ibrutinib, acalabrutinib, and zanubrutinib approved for chronic lymphocytic leukemia. Pirtobrutinib is a newer version of these BTK inhibitors. And, I will admit that I have been pessimistic over time. How many new BTK inhibitors do we actually need in the world? Why are we spending so much time on these drugs that are so similar to other drugs? This was a large international phase 3 trial comparing to BTK inhibitors, pirtobrutinib and ibrutinib. And I'll just highlight a couple of things. One of which is that you can see that in the ibrutinib arm, more people discontinued the treatment due to AE, which is an adverse event. So more people stopped the treatment due to poor tolerability when they were in the ibrutinib arm, as well as you can see there was a higher rate of progressive disease. So the ibrutinib arm appeared to be less well tolerated and less effective in this large phase 3 trial. And the results were very similar in the treatment naive population, as well as those people who had relapsed or refractory chronic lymphocytic leukemia. And again, you can see that something similar shown here with superior overall response rates. The interesting thing is that the response rates, although they're higher in ibrutinib, I don't think they tell the whole story for why there is an improvement in progression-free survival. I think some of that is simply put tolerability. When people are able to continue to take a drug, it tends to work better than if they have to stop it because of issues with tolerability. And so it does beg the question of how many of these drugs should we have and should we keep working on it? But I think in this case, it's the proof of the pudding is in the eating, and it's a good thing that scientists did continue to work on a new drug. The other interesting thing about this trial is even though it was equally effective in the treatment naive and the relapsed refractory population, the approval is expected to come only in the relapsed refractory population. They're not submitting for approval in the treatment naive population yet. And so if we want to have pirtobrutinib available to people with treatment naive chronic lymphocytic leukemia, we need to do it still in the context of clinical trials. And we're looking to bring one of those on board in the near future at NYU. I'll skip past this, but this essentially shows some of the nuisance type side effects that are more common with ibrutinib. Next slide. The last topic I'll touch on briefly are degraders. So for the longest time, we've had what we call inhibitors. So a protein binds to an enzyme and prevents that enzyme from working. Inevitably, enzymes somehow find ways around these inhibitors by acquiring certain mutations that prevent the inhibitors from binding or from working. And so a new concept that's come along is a way to actually degrade these proteins. In this case, the protein highlighted here is a protein called BCL6, which is active in multiple B-cell lymphomas. And by using this degrader, you can see on the far left-hand slide, there's this sort of barbell-shaped thing. One end of it hooks into the enzyme. The other end, left half of it binds to another protein that targets the BCL6 protein for total degradation. We've seen this process worked in the case of BTK inhibitors. So unlike zanubrutinib or pirtobrutinib, which just inhibit it, we've seen BTK degraders. And this was an interesting one targeting BCL6. I bring this up because one of our upcoming collaborators, Dr. Cerchietti, has really based a career on BCL6 early on, and we found that um, targets like BCL6, which are transcription regulators, can be very difficult to target, but the degraders suddenly make that possible. And in this very heavily treated population with follicular lymphoma and diffuse large B-cell lymphoma, including people with prior CAR T cells and other drugs. You can go to the next slide, please. Anyways, the treatment overall was very well tolerated with mostly low severity type side effects, which are treatment-related adverse events. You can see on the left-hand side, the bars are mostly green, which means low severity. And on the next slide, we'll have a look at the efficacy of the next slide, please. Have a look at the efficacy, which you can see is very high in follicular lymphoma, it's just an exciting era in medicine when targets that we previously thought were undruggable are suddenly druggable. And not only are they druggable, but they can be druggable to a great effect. So it's an exciting time. I think bispecific antibodies are clearly changing the therapeutic landscape of indolent non-Hodgkin's lymphomas. We have several upcoming trials at NYU, including the Cervatamig that I mentioned, mosunetuzumab. Sam mentioned a trial that's coming in follicular lymphoma. We have trials in epcoritamab and an anti-BAFF-R receptor bispecific antibody. Sam mentioned a CAR T cell there, but we also have an antibody there. I think many people with mantle cell lymphoma may never see chemotherapy going forward. We have so many good drugs there, but the duration of the therapy remains an open question that's being asked and hopefully answered by some ongoing and upcoming national trials that we'll have at NYU. And despite the efficacy and safety of a lot of new drugs, it's still clearly possible to develop new drugs that are even safer and better. I was a little bit pessimistic about this in the case of BTK inhibitors, but clearly I was wrong, and it's exciting to have these drugs moving forward. And lastly, new classes of drugs continue to emerge, despite the fact that we have so many effective and well-tolerated options. These new classes of drugs keep coming out, and there are multiple labs working on this at NYU. This is our lymphoma team here. I want to, highlight that it's a big team, but also that we cover a big part of New York with offices in Manhattan, Brooklyn, on Long Island, as well as other areas where people can have lab testing, imaging, and even some treatment throughout the city. And so I think we're well equipped to take care of people. And if anybody has any questions, we can pretty much get anybody in to see one of us within 1 or 2 days, almost guaranteed. Happy to help out in any way.
Great. Thank you very much. And before we finish, I want to really highlight our collaboration here with Blood Cancer United. And this has been a partnership that many of us have had over many years. It's a fabulous organization we work with on research, on patient education, and lots of other projects. And it's great to have Nicole Fortoso here with us, who is the Patient and Community Outreach Manager for BCU, to give us a brief update on some of the activities of the organization. Nicole, thanks for joining.
Thank you. As Dr. Leonard mentioned, the Leukemia Lymphoma Society is now Blood Cancer United. We changed our name to the public back in September. So excited to be here and share more about our resources. This is my contact information. If any of this information is helpful to you all or you want to learn more about it, please feel free to reach out. This is my cell and my email. So to talk a little bit about what we do, and if you haven't heard of Blood Cancer United, our mission is to cure blood cancer and improve the quality of life for all our patients and their families. This is just a screenshot of our new website, bloodcancerunited.org, where you can learn more about blood cancers, how to navigate your care, different resources, and also get involved and learn more about the research we have going on. I wanted to plug in this specifically. So this is extra resources that we have for healthcare professional education and support. Our blood cancer conference happening here in Times Square is actually registration just opened up today. This is a conference where we have 300, anywhere from 300 to 500 folks in a room, and we're able to, this is open to caregivers, patients, and also healthcare providers, and you just get a sense of community in the blood cancer community. So I highly recommend if you are free to register, and I can include the link later in the session. So these are really the experts. Our information specialists are where you can get the one-on-one support about your blood cancer journey. So they're there from the beginning, from when you first receive diagnosis all the way through treatment and remission. These are licensed social workers and nurse practitioners who are the experts and can answer any of your questions, and know the questions that you can ask your doctor as well. Another one of our resources we have is our Clinical Trial Support Center. So this is where you can work with the clinical trial nurse navigator one-on-one, and they'll help you find clinical trials and personally assist you through their process. If you've been told about some clinical trials through your doctor and your medical team, they can help you navigate and learn what questions to ask for your best treatment options. Another resource that we have at Blood Cancer United is our free nutritional consultations. These are 30-minute one-on-one nutritional consultations, and they provide you educational resources. One thing I like to note about our nutrition consultations they're free for all cancers, not just blood cancers. So if you know anyone with another type of cancer, this is available to them as well at no cost. I like to talk about our resources in three categories. Emotional is one of them, financial is the other, and then the third being educational. Our First Connection program is one of our peer-to-peer emotional support programs. This is where you're connected with someone with a similar diagnosis or a similar treatment plan. Anything that you're really looking for, we're able to connect you with someone, and these are trained volunteers who have been there and can share their experience. We find a lot of folks that they best connect with someone who's gone through a similar experience as them, and this is a way to get involved. Another one of our emotional support resources are our patient community. The best way to describe this is like a Facebook platform. You're able to connect and join different groups within the patient community. Some of these groups can be on people who prefer to have a positive outlook on their cancer journey, people who prefer to talk about what it's like to have a family unit, etc. But these are different groups, and they can talk about different diagnoses and also different walks of life through going through a cancer journey. Some of our support programs, we do have family support programs. These are open for patients and their families. And this is where they can go receive information, education, and feeling comfortable in an environment that provides care. As I mentioned, our online chats, this is a friendly forum where you can share experiences and chat with others about anything in the initial phase of a diagnosis. And then our caregiver support, we do offer many resources for caregivers as well, also free of charge. Some of our financial support programs are big to our patient aid program, which is a $100 stipend for eligible blood cancer patients, as well as our urgent need program. We also, in the next slide, talk about another one of our programs, but these programs are available on an open to close basis, and you're able to connect with our informational resource specialists in that first slide to talk about the options that you qualify for. Our medical debt case management program is a new one. We've relieved over $5 million of medical debt in the past year, and this is due to these one-on-one consultations with our medical debt case management team. They not only walk you through current medical debt, they also, one thing I like to mention is that they're able to help you walk you through preventing it as well. I know that it's the last thing on a lot of people's mind, but it's something that we see so often. How to prevent it would be a great active step in the beginning of your diagnosis. And also, they do help you fill out several forms, such as disability forms, unemployment forms, etc. These are some of the free educational booklet and fact sheets that we offer. These are free to order online. You're able to order them directly to your home and also download the free version online as well. These are other options that we also offer to learn more. As I mentioned, we have healthcare provider resources, but we also have educational booklets, fact sheets, webcast videos, and podcasts all available on our website. These are some ways to get involved with us. We're currently looking for volunteers to volunteer at our blood cancer conference on March 14th. So if this is a day that you're available and you would love to not only participate in our conference, but also volunteer, we would love to have you. And we also encourage folks to share their story and connect with us and join a campaign when possible.
We now have roughly 5 minutes left for questions, and we have 9 questions. So I'm going to ask our panelists to respond to questions in 30 seconds or less. And I'm going to start with Dr. Martin. How can I balance potential benefits of newer therapies with quality of life, especially for indolent lymphomas?
I think short story there is it's a conversation with your physician. There are, as you heard, more options than you can imagine. And I think having a detailed conversation with your doctor is probably the key to that, to find the treatment that's right for you. The other thing that I would not overlook are clinical trials. Many of the drugs that are coming in clinical trials can be expected to be better tolerated than some of the historical agents. So just because it's an indolent lymphoma, it doesn't mean we shouldn't be exploring some of these clinical trial options that I think are potentially really promising.
Great. Dr. Yamshon, what's the average durability of CAR T cell therapy for treating diffuse large B cell lymphoma? Meaning how long does the treatment last if it works in a patient?
Yeah, so just quickly, we measure the efficacy of treatments for lymphoma and for cancer in general in a few different ways. So the first is how many patients respond, and then we also kind of measure how long that lasts. And we think of CAR T cell therapy for the patients who have complete remissions as what we call a curative intent therapy, meaning that the goal for diffuse large B cell lymphoma is that it stays away forever. And I counsel my patients who come in for CAR T that my hope is that it will cure them and keep their lymphoma away for good. So obviously not everyone gets there, but that I think is the goal. So I would say it's tough um, to every patient's different, but the goal is that it is a cure
Right.
in diffuse large B cell lymphoma.
Great. Dr. Diefenbach, where can patients learn more about clinical trials related to these findings and their eligibility? And maybe you can mention at NYU how people can learn more.
So there are many ways to learn about clinical trials. Probably the best way is to ask your doctor. You can also email any of us from the call tonight, and we're all happy to tell you about clinical trials at NYU. You can go on the NYU website and look at the clinical trials that are available at NYU. You can reach out to places like Blood Cancer United, who can also connect you with doctors who can tell you about clinical trials. And finally, if you have a very, very rare cancer and you're interested in clinical trials across the country, there's a website called clinicaltrials.gov where you can look up any cancer condition there is and find clinical trials that are recruiting in that area. But I would say the short answer is if you have lymphoma and you're interested in a lymphoma clinical trial, you've just met three of us, actually four of us, including Dr. Leonard, on the call tonight. The Blood Cancer United has all of our contact information if you weren't able to get it from the chat or from our slides. And you can reach out to us at any time, and we're happy to walk you through the clinical trials that we have here at NYU.
Great. Questions about follow-up care and monitoring, and I'm just going to answer this one briefly, that it really depends on your situation. If you have a lymphoma that is at a point where it's not likely to come back, if it's one of the lymphomas that's curable and you've gone a long time and it hasn't come back, that follow-up care is one set of issues. Whereas if you have a lymphoma that's likely to come back over time, particularly some of the indolent lymphomas, then you need to keep maybe a little bit closer eye on it. So it really is individualized. And I think talk to your doctor about what your expectations are and what you need to watch for. The other thing is that certain therapies have long-term side effects that you have to keep an eye out for as well. That can be on the blood counts, on the immune system, on the heart, other things. So it really is something you need to talk about with your doctor. Dr. Yamshan, what are the developments for patients who have relapsed after a previous CAR T? What do you do if someone has a CAR T? And as you alluded to, there are a fair number of people, unfortunately, where the CAR T, is temporary or doesn't work. What are some of the things you think about there briefly?
Yeah, so currently the standard is to move on to another type of therapy. And actually, many of the therapies that we discussed, in particular the bispecific antibodies, have been shown to be remarkably effective even after someone's lymphoma comes back after CAR T. But many of the strategies that I was discussing tonight in terms of improving CAR T are also being used to give CAR T after an initial CAR T doesn't work. So, for example, many of those CAR T products that target a new spot on the lymphoma cells can be given even after an initial CAR T doesn't work. But also some of those ones that even target the same spot but try to enhance the CAR T cells, durability and ability to keep from getting exhausted, are used. And actually, many of our CAR T cell trials that we have open here also do enroll people who have gotten a previous standard CAR T. So many different options, including CAR after CAR.
Right. So lymphoma is diagnosed typically through a biopsy. That was a question that came up. It really has to be directed typically by a hematologist. Most commonly, it presents with a lump somewhere, then a large lymph node. And the other question here before we finish up is around transformation. That's when an indolent or slow-growing lymphoma changes to be more aggressive. That happens over many years in perhaps 10 to 20% of people. And if that has to be diagnosed also by a biopsy, it tends to be suspected when there's a rapid change in what's going on in the lymphoma, or if it's resistant to treatment, if it's an indolent type that typically responds well. So sorry for the short answers for some of these. These are great questions. Before we wrap up, I want to turn it back over to Katie Grace just for some final comments. Thank you all.